SNPs as physical markers. Single nucleotide polymorphisms, or SNPs, commonly pronounced as SNPs. They are single base pair positions in a genomic DNA at which different sequence alternatives that may be called as alleles, they may exist in a population. कि कोई भी डीएनए का सेगमेंट जो कि एक इंडिविजुअल से दूसरे इंडिविजुअल में प्रेजेंट होता है उसमें सिंगल बेस पेयर जो है वो वेरी करती है और इन जो सिंगल बेस पेयर की जो वेरिएशन होती है विद इन अ पर्टिकुलर डीएनए सेगमेंट इधर इट इज़ अ जीन और एनी अदर सेगमेंट they are commonly called as SNPs or single nucleotide polymorphisms. Now, in routine life, organisms they may be exposed to different environmental factors or chemical mutagens that may introduce base pair changes that are commonly called as mutations. In order to differentiate such rare mutations from SNPs, it has been suggested that the allele frequency it must be 1% or higher ताकि इसको जो रेयर म्यूटेशंस होती हैं उससे इसको डिफरेंशिएट किया जा सके. Now different type of nucleotide variations they can be considered as SNPs or SNPs because most of the time this term it is used in loose sense. For example, in case of cDNA, the single base pair variation, they may be called as C SNPs because it has been suggested that they are showing the underlying genomic DNA sequence. In the same way, if some addition or deletion is there, that are commonly called as indels, that may be the result of addition of the base or removal of the base that are called as indels. They can also be regarded as SNPs. And the most common type that is detected by restriction fragment length polymorphism, they are called as the SNPs. If you remember, we discussed that the 0.1% genome is variable from one human individual to another, and these small variations they can influence the target sites for different type of restriction enzymes, so that after digestion with particular restriction enzyme we will get a particular or characteristic bending pattern, so that fragments they will be separated depending upon their lengths on agarose gel electrophoresis. There are some other instances where two alleles they can be differentiated on the basis of their phenotype. इसमें जो डिजीज़ है जो कि आप मैट्रिक से या एफएससी के कोर्सेज से भी पढ़ते आए हैं कि सिकल सेल एनीमिया, इसमें आपको याद हो कि देयर इज़ ओनली वन सब्स्टिट्यूशन ऑफ द अमीनो एसिड कि जिसमें जो ग्लूटामीन है इट इज़ रिप्लेस्ड विथ वेलिन because here the one nucleotide base pair is replaced with another and it can change the sequence of the triplet code, and in place of glutamic acid there is valine and a disease is resulted which is called as sickle cell anemia. So it is not going to influence the phenotype of a particular trait, but also it can change the restriction site of the enzyme के सेम साइट जो है एक न्यूक्लियोटाइड के चेंज होने की वजह से एक एंजाइम की रिस्ट्रिक्शन साइट भी चेंज हो जाती है. सो वी कैन एक्सप्लेन इट विद द हेल्प ऑफ अ डायग्राम. So this is a sequence that is showing single base pair substitution, and in the upper strand here TA base pair, especially in upper strand like thymine, three different possibilities are there that this thymine it may be replaced with either cytosine adenine or guanine. And if it is replaced with another base, like here, suppose a central one, the enzyme site, which is called as MST2 restriction enzyme, its site will be influenced. Like here in the lower diagram, which is diagrammed in a circle, a 3 amino acid sequence has been shown, like proline, then glutamic acid, and here A is replaced with T. Here in the lower strand, T is there in place of A, so that now in place of glutamic acid there is valine in the center. So you can say that the beta globin protein, it is now changed in sequence that can result in sickle cell anemia. SNPs, they are very good physical markers for the mapping. The reason is that they may be present in high density. अगर हम ह्यूमन जीनोम को कंसिडर करें तो हर 1000 न्यूक्लियोटाइड्स के बाद एक SNPs you can expect. So in this way, it has been reported that there may be 3 million such cases, and even now more reports are there that have increased the number of SNPs. So because of their density, they are good markers for genome mapping. Now different methods have been used to detect single nucleotide polymorphism that may be based on enzymatic reaction, where we can use different PCR reactions. We are again going to discuss again and again some PCR techniques that we have already discussed that may be related to RFLP, AFLP, rapid, that is randomly amplified polymorphic DNA, and some other PCR methods. And then some electrophoretic method can be used where the DNA fragments, they will be isolated on the basis of their length. And some chromatographic technique where the DNA molecules, they will be separated either depending upon their melting temperature. And here the most common technique that is used, it is denaturing high performance liquid chromatography, or DHPLC. And here, if single base pair substitution is there in the double-stranded DNA molecule, that may vary from one individual to another. Although some other methods are also available, and here liquid chromatographic technique can be used. So if two DNA molecules, they are having the same length, same number of nucleotides, even then after denaturation they can be separated. On what basis? On the basis of their melting temperature. As is the character of SNPs, that there is single base pair variation in a particular gene or genomic DNA segment. If one base, it is changed, it will also influence the melting temperature, so that two DNA molecules, they will be differentiated. So SNPs, they are good markers for genome mapping.